Girl, I'm calling, can you pick up? It's been a long while since I put up. You made me feel like everything I've done, all the things we did.
Alright, so welcome once again to my channel. As you can see, I'm Isaac by name. Yeah, but my channel name is Jellyville, so make sure you subscribe to my channel for more of your studies. This is Biochemistry, and I'm come to discuss cell fractionation with you. So when we are talking about cell fractionation, it's all about separating the components of a cell. Understand? A cell may be made up of a nucleus, cell membrane, cytoplasm, and all those stuffs. You can separate each of the components, okay, into different test tubes with a process called cell fractionation. Biochemists work with cell extracts, okay. They work with the cell components. They don't work with the whole cell. They work with the component that they want to work with. So let's say if they want to work with nucleus, they work with the nucleus of a cell. They don't work with the whole cell. Now, when we are talking about this extract, before you obtain this extract, there are several processes that you should go through. And these several processes are grouped into two main steps, which one is homogenization, and the other main step too is differential centrifuge. Okay, so the homogenization entails a whole lot. This is where you are going to take the cell and split it up into its components. But how are you going to do it? We all know that cell is very small. So in terms of the homogenization, we can take the first step to be through grinding. The grinding may be, let's say, there is a blender which is scientific blender. That is what the scientists use through grinding. So the grinding is all about splitting the components of the cell, okay, in a sample. So let's say you have the sample, you have your sample, this is your sample, and you place it in the blender or the scientific blender or the grinder. But let's call this one blender because to make communication very effective, let's call this one blender. So if you pour the sample into it, you have a whole lot of cells in that sample. Now what do you have to do in order to obtain the samples? You have to grind it. So from here, you can grind your sample using the scientific blender and later transfer it into your test tube or your container that you are actually using to perform your experiment. So after getting your grinded sample, the sample is no longer called sample. I don't know whether you get what I'm trying to say, but your sample in the blender will no more be called sample, but rather be called homogenate. And it's called homogenate because it contains all the cell components are now being released into the sample. So now your homogenate, it has to be purified before it undergoes the second process, which is called the differential centrifuge. So from here, you have to undergo a process called filtration. So you can undergo a filtration process in order to purify your sample or your homogenate. The reason why we undergo filtration or we undergo this purification is to remove any cellular debris. For instance, as we use the blender, we were able to grind the cell. After grinding the cell, we are going to get some cell debris, like let's say the cell membrane. The cell membrane, they are things that we actually don't need in our experiment, or we don't need it as part of our cell extract. For that matter, we need to purify it. Purifying it using filtration will enable you to identify your components or your extract. So the main process or the main aim of doing the purification is to remove cell debris. Alright, so from here, you transfer the homogenate into a test tube. So this is called the homogenate, and it contains all the cell components. It has been released into the sample. Now, before you undergo your centrifuge, make sure that you have all your homogenates in one test tube. Now, you have to insert. A centrifuge is like a machine. It's a scientific machine where we have a whole lot of slots that fit into each of the test tube depending on the sizes. So we fit the test tube containing the homogenate into the centrifuge. What the centrifuge is going to do is to is to spin around. So the centrifuge will spin with a test tube inside the centrifuge. The centrifuge is going to spin around. So one thing you should notice is that all the cell components have different masses. That's what you should keep in mind. So this means that you don't have to start your centrifugation with a very high speed. You should move slow. So the more you are going, or the more you are separating your extracts, the faster you'll be going. So at the initial stage, you have to be slow, or you have to move with a very low velocity or speed. So after your first spin, you are going to get the extracts that are very dense. Okay, the extracts that are very dense will be the one that will be extracted first. So they rather be called pellets, and that pellet could be any of the extracts that you see to be very more dense. So let's say a cell, for instance, the most dense component of a cell is, let's say, the nuclei or the nucleus. So that one will first be extracted. So when it's first extracted, it will settle at the bottom like a sediment. It will settle at the bottom. Now it is called pellets because it contains the pellet is actually the sample or the extract that has been separated from the supernatant. The supernatant are still, the homogenate, or the supernatant are still let's say the fluid or the sample that still contains some of the cell components. It contains a mixture of the cell components. So after separating your nucleus, you know that a cell doesn't contain only nucleus. It can contain ribosomes, can contain cytoplasm, it can contain, let's say, endoplasmic reticulum and all those stuff. So they may still be in the supernatant. Now you move to the second process. You are going to transfer your supernatant into another test tube in order to separate the nucleus fully from the supernatant. You need to transfer the supernatant, which is a fluid, into another test tube. Now you've transferred the supernatant into another test tube. It undergoes another spin again, or centrifugation. So I've used this arrow to show that it has undergone another centrifugation again. Now when it undergoes centrifugation, this time the speed will be greater than the one that we used initially. So let's say for about 10 minutes, we are going to get another pellet again, and this pellet may be containing mitochondria, or let's say if you are dealing with plant cell, mitochondria and chloroplast are having similar masses. So in this situation, if you are dealing with plant cell, this pellet may contain both mitochondria and chloroplast, but that doesn't mean that they cannot be separated. They can still be separated. So when we get there, I'll let you know how they are still the chloroplast and the mitochondria can still be separated. Now this process continues. So you have another supernatant again. You transfer it into another test tube. Then you put it in the centrifuge or the differential centrifuge. You spin it again. You are going to get another supernatant with a pellet. That pellet may contain, let's say, the less dense components apart from mitochondria and chloroplast, may be the Golgi body. Golgi body or endoplasmic reticulum. So they are the things that actually have similar size too. So that pellet that you'll be having, the next pellet you'll be having may contain either endoplasmic reticulum or Golgi body. So from there, since they also have similar masses, you can also keep that test tube too and label it, let's say, Golgi body plus something like endoplasmic reticulum. Then you transfer the supernatant again. So from there, this means that as you continue transferring your supernatant into different test tubes for the centrifugation process to continue, what you have to note is that the speed should be increasing. As you can see, there were differential. It differentiates the components according to their mass or their weight. So the more you increase the speed of the centrifuge, the more you are going to obtain your extracts. Now, this process is achieved according to their weight, as I've stated earlier, or their mass, or the mass of the cell components. Now let's delve into it very well. So as you can see, when you're having a cell, we have nuclei, but nuclei, this one just show the masses. So nuclei is greater than what? Mitochondria or chloroplast. The same way the mitochondria or the chloroplast is greater than what? The endoplasmic reticulum or the Golgi body. And the same way those two are also greater than what? The mass of what? Ribosome. Now let's delve into it. Now that we have our pellet that contains, let's say, mitochondria or chloroplast, those two cell components have, similar masses. But that doesn't mean that they cannot be separated. They can be separated using a method called what? Density gradient centrifugation. And the gadget or the equipment that is used in separating those two components with similar masses is called the density gradient centrifuge. Now, this is used to separate cellular components that have similar masses and cannot be separated with the differential or centrifuge. So if there is something that the differential centrifuge cannot separate it according to their masses, then probably you should go for the density gradient centrifuge. That one will also help you separate the components irrespective of their masses. Okay, so there's one thing I forgot to also mention about, which is the grinding, the homogenization. This thing we can also use the chemical or the ultrasonics. If you don't have the blender, if you don't have the scientific blender and you want to go the other way around using the chemicals, you can also use, when you're using chemicals like, let's say, there are some enzymes or chemicals that are actually respective in their works, or they are specific in their works by breaking down, let's say, the cell membrane. Some chemicals are there that have specification in breaking down cell membrane. So when you add that chemical into your sample, it's going to break down all the cell membranes. Okay, and you know that the cell membrane serves as a boundary for the cell components and the environment. So if the cell membrane is broken, definitely the cell is going to release what? Its components. The same way you can also use the ultrasonics. The ultrasonics, they are actually, or something like ultrasonic homogenization, it actually involves what? The use of high frequency. Okay, when you're using high frequency sounds, like let's say 20 to 40 kilohertz, it's going to disrupt the cell membrane. It's going to, because you know that sounds are made up of waves, and waves can cause interferences. So it can actually disrupt the cell membrane using some mechanical forces, and thereby it will release the cell components. That's how either chemical or ultrasonics works. So that one you should keep in mind, because it's also a step that is used during homogenization when we are not having the scientific blender around, or you cannot use the grinding process. Now, let's talk about enzyme makers. When you are talking about enzyme makers, these makers, enzyme markers. When I'm talking about these enzyme markers, they are actually specific in their action, because we all know enzyme. Like sometimes when you talk about, let's say, stereochemistry, for instance, we have enzymes. Enzymes are specific in their orientation. Let's say when the enantiomer that is coming is having a different orientation as compared to the enzyme, or as compared to how the enzyme is designed or folded, it's actually not going towards. There will be no reaction, or there will be no interaction, because the enzyme does not have the same orientation as the enantiomer or the molecule that is coming. So the same way as we apply here, so the enzyme actually serves as indicators for the presence of activities of a cell. Let's say you are having a sample. It has undergone homogenization, but you don't know whether actually the cell is having nucleus or is having mitochondria or something. You can use the enzyme markers. Enzyme markers, they are different types of enzyme markers, and these enzyme markers, depending on what you use and depending on what you are actually looking for in the sample. Let's say you want to look out for, mitochondria. There is an enzyme marker called, let's say, cytochrome, and this cytochrome is going to help you if you apply this cytochrome or this enzyme into the sample and you don't see any interaction. This means that what you are looking for. It's not there, but if it is there, if your mitochondria is there, there will be an interaction that will show that yes, this marker has actually identified, let's say, mitochondria, because it will be able to interact with it, because how the enzyme has been designed will be able, or it's designed to actually, react with mitochondria. So the moment you add that marker to it, it's going to identify whether there's a presence of this particular cell component that you are looking for. All right. So this is all about cell homogenization. So make sure you subscribe to my channel. Anything that's bothering you, you can, type it in my comment section. I'll go and look at it and make a video on it, and I'll make sure that you understand it very well. Don't forget to subscribe to my channel, and thank you very much for watching my video. Like and comment. Thank you.
Surviving is winning, Franklin. Everything else is bullshit. Fairy tales spun by people afraid to look life in the eye.